All right, we are live.
Okay. Well, welcome everybody to our workshop. I want to thank everybody for joining us for our first virtual version of the CNB workshop. And the theme of this workshop is integrative muscle modeling for neuromechanics. We're trying to bring together people who are interested in experimental approaches to understanding muscle structure and function, as well as computational approaches, so that we can talk about integrating between experimental and computational approaches, and also integrating across scales from molecular understanding of muscle biophysics all the way up to organismal movement and human-machine interaction. So first, I want to just give a little bit of background about the goals of the Comparative Neuromuscular Biomechanics group, and then we'll get started with our first set of keynote speakers. So the Comparative Neuromuscular Biomechanics group was started a couple of years ago, but last year at the Integrative Society of Biomechanics, the ISB-ASB meeting in Calgary, we officially became a working group of the International Society of Biomechanics. And I'd like to really thank Dr. Jonas Rubinson for his initiative in starting the group and bringing us together. The goal is to facilitate more collaboration and dialogue between human-focused biomechanics and the comparative biomechanics field. Traditionally, the comparative biomechanics has its home in societies like the Society for Experimental Biology and the Society for Integrative and Comparative Biomechanics. And meetings like ISB and ASB typically are mostly focused on human biomechanics with relatively little comparative participation. So we'd really like to get people talking between these fields more. And we think that there's a lot of opportunity for this cross collaboration between these fields to help us integrate across scales in our understanding of muscle function, from molecular mechanisms up to whole organism movement. And right now we've been operating with an interim board. So I'd like to thank all of the board members for their contributions to organizing this workshop, as well as running the working group. So the goal of this workshop is to bring us all together just to talk about these issues and really try to shape our activities as we go forward as a group, because we really want this to be a community-led effort. So we're bringing people who we believe are working at this interface and have interest in this cross-disciplinary interaction that we've set out. So we've handpicked all of the participants of the brainstorming sessions because we believe that you have something to contribute in shaping the way we move forward as a group. So one of the big focuses here, we'd like to talk about how we can better integrate between experimental approaches and computational approaches. And one of the things that we would really like to see is open sharing of muscle data sets, models, and simulation tools so that we can better progress as a field by sharing these resources. And as I said before, that the goal overall is to try to facilitate integration across organizational scales. So this workshop is just a prelude to what we hope will be an in-person meeting at ISB 2021, and we've received a small grant from the Company of Biologists to sponsor this meeting, and we will use that grant to help facilitate participation of early career researchers. This meeting will also have an open call for poster contributions, so there will be wider and we hope larger opportunity for participation from anyone around the world to contribute, because we will also hold both in-person and virtual components to that workshop as well. So before we get started, I won't talk for much longer, but this all started on the sort of back of a napkin conversation over beers at ISB in Calgary. So I'd like to just thank Greg for helping me to pull this together, this vision that we had over a year ago. And I'll kick it off by just reminding everybody of the schedule. We'll have our first set of keynotes by Simon Sponberg and Natalie Holt, talking about experimental approaches and the challenges of integrating across organizational scales. And then we will break out into our first brainstorming session on that theme. After the keynotes, I'll come back and just remind everybody about the instructions on the breakout sessions. So without further ado, I'd like to let Simon and Natalie take the stage. Thanks.
Guess I'm starting. Good morning, everyone. Okay, let me share my screen. Everyone see a title slide okay? Thank you. That was a lot like class, just blank faces. Morning, everyone. I'm super excited to be here and see you all, at least virtually. I really wish we could see each other in person, but that's not happening. Thank you so much to Monica and Greg for the highly unenviable task of trying to organize an online meeting. I'm glad I'm not doing it. Sorry. So I'm super excited to be here and talking with Simon. We've had some really nice discussions about this. And we're going to talk today about experimental approaches and the challenges of integrating across spatial scales from molecules to whole animals. And we entitled our talk The Allure of Muscle. In our early discussions, we kept coming back to this point that we really love working on muscle because it seems tangible. It really seems like it should be simple enough that we can understand it, and yet we just don't. And so it kind of keeps dragging us back into these questions that we just can't quite seem to get a handle on. My slides are not moving, so that's fun. There we go. Okay, so skeletal muscle is our biological motor. It drives most of organismal movement. And this is really grounded in the interactions between the contractile proteins actin and myosin. And we think of this kind of very simple cross-bridge cycle where myosin heads consume ATP and they use this to generate force and do work in interaction with actin. So this is kind of our base level of where muscle operates. And we've really used some very kind of static isometric and isotonic experiments, so the force length and force velocity relationship, as support for this kind of simple cross-bridge theory. So we've got this idea that static properties support muscle operating via this simple cross-bridge model. But what's really interesting to, I think, both Simon and I is the huge spatial scale over which muscle operates. So if we think about individual myosin heads, they undergo a cross-bridge cycle. They move on the order of 8 nanometers. It's this tiny molecular scale movement. And somehow the integration of many of these cross-bridge cycles powers organismal movement with limb movements on the order of centimeters and whole organism movement of much larger. And so we've got this huge spatial scale to span. And within this, we have an extraordinary amount of structural complexity. So actin and myosin are organized into these filamentous proteins. These proteins are not rigid. They exhibit some small degree of compliance. This compliance can change cross-bridge binding probability as muscles are activated and generate force and strain these filaments. So we're no longer thinking about one cross-bridge as an independent actuator, but more as something that can have cooperative binding based on the strain that it's experiencing. And this has implications for function and force generation under different circumstances. We have these filamentous proteins organized into sarcomeres. We generally depict this in 2D, but this is actually really a 3D lattice, with the lattice spacing having important implications again for binding probability and force. Even in 2D, our sarcomere is nowhere near as simple as actin and myosin. We have this extensive network of cytoskeletal proteins. Maybe most notably titin, which has been considered to be a third contractile protein changing in response to activation. We've also got a bunch of other stuff. We've got myosin binding protein C that's kind of now maybe the fourth filament. We have a lot happening and we have a very crowded environment. So we've got this complicated sarcomeric environment. We organize these sarcomeres end-to-end in series in myofibrils and package these into muscle fibers or cells. And we surround these muscle fibers with a connective tissue extracellular matrix that we can see a nice image from David Sloboda here in cross-section. So we're essentially working with something that is a fluid-filled bag wrapped in this collagenous structure. And so as we try and deform this, it's going to resist deformation, it's going to experience fluid pressure, and there's all kind of complex material properties happening. We organize these fibers into bundles of muscle fibers. We surround these with more connective tissue. We organize these bundles of fibers with some geometry into a whole muscle. And we see kind of connective tissue aponeurosis sheets and long tendons connecting to these. And at this scale, we also have important 3D considerations. So much like at the lattice, we've got a whole muscle operating in 3 dimensions. It's got asymmetries in its fiber orientation and its connective tissue placement. And the way in which this muscle changes shape as it contracts is going to have big implications for its performance. We then put these muscles into a whole organism. We have a highly redundant muscle system. So we've got many muscles spanning each joint. We've got the nervous system controlling this whole thing. This is kind of where I tap out once we get to nervous system out. And then we've got the muscles interacting with environmental forces. So the muscles are acting to move the body, and we're then experiencing forces that the muscles are subject to. So we've got this huge amount of spatial scale and structural complexity going on within muscle. And I think this really kind of contributes to our limitations in our understanding. So we started with this basic cross-bridge theory, myosin heads acting as independent actuators, and these static force length and force velocity properties. But we have a huge amount of evidence now that if we take these properties and put them into simple hill-type muscles, we get a horrible fit of the predictions of those models to a lot of experimental data. And it just doesn't do a good job. Those static properties don't well explain muscle performance during movement. And we have some ideas of why. So even in relatively static conditions, we have things like history dependence. If we allow muscle to shorten before doing an isometric contraction, it doesn't generate as much force. If we stretch it, it generates more force. Those force length and force velocity relationships move around all over the place with things like activation and contractile history. So even under relatively static conditions like we see here, we see pretty big deviations from this basic cross-bridge theory. And it just all goes to shit once we start looking at an organism moving. And so what it really appears is though this kind of basic cross-bridge model works well in very static, very controlled conditions. But all of this spatial scale and structural complexity becomes much more important once we start thinking about real movements, which is where we want to understand muscle. And so in Simon and I's discussion, we kind of came up with 4 experimental areas that either we're working on or thinking about as a way of how are we trying to get at understanding this spatial scale and structural complexity. And this is by no means exhaustive. We kind of came up with a giant list and then decided we couldn't deal with the giant list. So we're not talking about everything. This is really a start point as to where our thinking is to hopefully get discussion going. So we started thinking about simultaneous multi-scale experiments. How do we do experiments that capture what is happening across multiple levels of muscle? Simultaneous is ideal. I might also argue that collaboration that does compatible levels of muscle experiments across different spatial scales would also be helpful. We're talking about doing muscle experiments either in natural conditions with unsteady environments or that replicate that. And Simon is going to talk about these 2 later. And I want to talk about 2 areas that I've been working on, which is looking at varying muscle activation. And then maybe a kind of future direction for me, thinking more about comparative approaches that felt like it might be useful for this group. And I'll talk a little bit about why these 2 approaches are helpful. So if we start with muscle activation, I've been working on this for a while and really kind of feel like it's a good way of understanding some of this complexity of structure across muscle. So organisms vary activation in vivo. This is a bird in flight. It's going to vary the EMG or electrical activity of its muscle to change speed, to change performance, all controlled by activation. We can change activation in vivo by varying motor unit recruitment, so by varying the number of muscle fibers that are active. And we can change it by rate coding, so changing the frequency of these action potentials that's going to the muscle. And in doing this, what we're essentially doing is we're changing the relationship between the number of active cross bridges. So as we turn down activation by either of these means, we have less cross bridges cycling, but we still have an intact muscle. So we're changing the relationship between cross bridge cycling and structure if structure has an effect, that should show up in non-linearities of activation on muscle performance. So it's this kind of nice way of keeping all of these complex bits of structure in their correct geometric and spatial arrangement and having some perturbation that lets us investigate that. And so we can replicate this in vitro or in situ by changing the amplitude of stimulation or by changing the frequency of stimulation. There's some very old techniques looking at nerve splitting and rotation of stimulation that I'd like to get back into. There's some nice new optogenetic models whereby we can use light to selectively activate different muscle fibers and give us a more physiological recruitment. But it's kind of a nice base technique. So I just want to talk briefly about one study that I've done that I think gives us an example of how this can help us understand muscle performance. And this was a study we did looking at the effect of recruiting different types of muscle fibers. So skeletal muscle is heterogeneous with respect to fiber type. Muscle fiber types vary from slow type 1 fibers to fast type 2B fibers. A lot of this variation is driven by the speed of myosin kinetics, basically. And they also have changes in metabolism and kinds of other things. But for this purpose, mostly speed. So in a rat plantaris muscle, we activated either all of the motor units and all of the muscle fibers, just the fast or just the slow ones using some selective activation protocols. And we looked at the effect of this activation on the force-velocity relationship. So this is just showing shortening velocity against relative force. And when we activate all of our motor units in the purple, we see a classic isotonic force-velocity relationship. When we activate either just the fastest motor units or just the slowest motor units, we see a pretty big drop in maximum shortening velocity. And this was super surprising because based on our simple cross-bridge model, we'd expect that if we have a fast myosin in our fast fibers, we'd see a high shortening velocity, and that's just not the case. And so our very speculative prediction from this was that maybe it's structural elements of muscle. Maybe it's this fact that fibers are essentially fluid-filled, fiber-wound cylinders with resistance to deformation that does this. And when we turn down the activation, we reduce the ability of cross bridges to deform the material properties of muscle, and particularly to deform them quickly. And so we get a reduction in shortening velocity. So it gave us kind of some insight into how some of these structural levels of muscle could be interacting with contractile proteins in order to determine function. I think we can go beyond this. This is a nice kind of start point. I've done a few experiments like this, but maybe one of the ways I'd like to take this in the future is looking at these different methods of activation. So changing number of active fibers via motor unit recruitment, or changing contraction, changing activation frequency. These have a really nice kind of pairing of ways of doing things. So we can make a muscle maximally contract and generate some force. We can turn down the activation and we can get a big drop in force by either of these methods. So we're controlling for what the whole muscle's doing. The whole muscle is generating the same amount of force, but we have it distributed very differently. If we change motor unit recruitment, we just have a few highly active fibers. Some fibers are generating a lot of force. If we turn down frequency, all fibers are generating a small amount of force. And that might seem kind of trivial, but I think it's really important for untangling some of these molecular mechanisms. If I want to know what deformation of the contractile proteins is doing to whole muscle function, if I maximally activate a few fibers, their contractile proteins are going to deform a lot. And we can also use this to maybe investigate titin. And I think some work out of Daniel Hahn's lab has been looking at doing this. If I turn on all my fibers, titin in every fiber should be doing whatever it's doing. If I only turn on a few fibers, I have much less titin contribution, but maybe more compliance of filament contributions. So I think it's a nice way to start parsing some of these complicated molecular mechanisms. We shift now to just briefly thinking about comparative approaches. We have a long history of comparative physiology. That was certainly why I started as a biologist. I was super interested in organismal diversity. If we go back to the late 1920s and look at the Crow principle, which says that For any problem that we want to understand in physiology, there'll be a best animal in which to study it. And in muscle, this has really looked like taking extreme examples of organismal performance, like frog jumping or sound production in rattlesnake tail shaker muscles or insect flight, and looking at how a single muscle can achieve this kind of one big function. And we've seen this demonstrate elastic mechanisms. We've seen it demonstrate large amounts of sarcoplasmic reticulum and very fast calcium kinetics. We've seen it demonstrate asynchrony between muscle contraction and nerve input. But what I really want to do is take this comparative approach and think about how we can apply it to this problem of spatial scales. And the way in which I think we can extend this approach is to start thinking about systematic differences in things that we're interested in across organisms. So for example, we have all of these phenomena like history dependence that we don't particularly understand. Do these vary across organisms, and what can that tell us about their mechanism? And also, how does muscle structure vary between organisms? So at the molecular and the bigger scale, can we leverage differences between organisms to understand what those structures are doing? So we take history dependence, this idea of if we stretch the muscle, we get more force out after we've stretched. We see it across vertebrates. We've been arguing bitterly about it for years and don't seem to have a resolved mechanism. But we see elements of this phenomenon in lots of other organisms. So we see stretch activation in insect asynchronous flight muscle, whereby if you stretch a muscle, you get this delayed rise in activation and force. We also see this to some extent in cardiac muscle. We see this, that I'm amazed no one studies more, this property of catch in mollusk adductor muscles, whereby even after calcium declines, you can get this prolonged holding of force very cheaply. And so we have these kind of things that aren't quite force enhancement, but they look a little bit like it. And we can maybe use some molecular differences between these organisms or tissues and our kind of standard vertebrate model to understand this. So we have variation in troponin that's thought to drive stretch activation. We have huge variation in, this is the length and structure of the titin molecules across vertebrates and invertebrates. So if I have catch in a mollusk muscle, what does its invertebrate titin-like molecule look like? And can that help me understand what it does in a vertebrate? We also could talk about muscle compliance in this sense. I just want to skip over this kind of quickly. So we have a lot of variation in sarcomere length across kind of disparate individuals. So from vertebrates through insects and crabs and mantis shrimp, we have this huge variation in sarcomere length that relates to a difference in contractile protein length. Everything we know about contractile protein compliance comes from, I think, rabbits and frogs. So who knows what these long sarcomeres in mantis shrimp, who knows what their contractile proteins look like? And maybe we can use that to probe what's the effect of compliance in those structures. We can do this at a bigger scale. So if we think about compliance in connective tissues, we know this varies between mammals and frogs. Maybe we can use this in the same way as we've used aging and pathology to understand the control of connective tissue shape change and start to get some kind of greater insight into that in a healthy system. And maybe we can do this in a way that's somewhat more phylogenetically rigorous. So if you look at our fast muscles, we have an insect flight muscle, a toadfish, and a rattlesnake, which has no phylogenetic bearing on each other. Same for sarcomere lengths, we're kind of all over the map. I think if we do this in a more rigorous way, we can start to look at intermediate states and so get at better mechanisms. And so I've really focused on these kind of naturally occurring perturbations in terms of changing activation level and looking at organismal diversity. I really like these approaches because at least up to the level of the muscle, you keep everything stuck together. You keep all of this structural complexity in its correct arrangement. You don't take everything apart and take a very reductive approach. And so it lets you see how things are functioning in a muscle as a complete entity. The problem with that is you get to the end of an experiment and you're like, cool, I did not expect to see that. Something is important, but we don't have a good sense of what. So I've spent maybe the last 5 years being like, this is wrong, but I don't have a better solution for you. Sorry, but we should look at that because it's cool. So I think that's where the strength of these experiments really lie, is that they highlight areas where our understanding isn't good and suggest mechanisms. They're also nice because they teach us about in vivo organismal performance, but I think it's really useful to have a back and forth between these kind of basically faking looking across different spatial scales experiments with some of the more kind of tangible, how do we actually do experiments at different scales, and how do we use the influence of the environment on that? So I think there's a lot of scope for moving back and forth. So with that, I'll hand over to Simon.
Okay, just switching over screens. If I can get a thumbs up that folks can see that slide fully. Okay, good. So we'll take over. It's always hard to do the tag team, especially virtually. Thanks, Natalie. So the sort of third approach that I want to start talking about is this transition that we've had in looking at the physiological level to really trying to consider unsteady conditions. And these are ones that arise when animals are perturbed, whether that's in fluids or on a more terrestrial scale. So just to go back and recap, we had, Natalie had talked about how we have used these sort of quasi-static measurements, the force length, force velocity curve, to characterize performance for a long time. And that really goes back, I mean, that was the right experiment to do to test the sliding filament hypothesis, right? We have to remember that these experiments were originally designed to test the underlying biophysical mechanism of it in a certain very particular context. They don't necessarily relate to the physiological performance that some of us are interested in. Another good example of that is sort of the small scale sinusoidal analysis that Machen and Pringle and then later Kwai and Brandt did, where you do these really tiny little oscillations in strain of a muscle, and then you sweep through frequency. And this gives you a linear approximation of muscle's behavior as a material. And again, this isn't very physiologically relevant, but you can actually fit the Nyquist plot, the bottom plot down here, to 3 different time constants that relate to the binding kinetics of myosin. So again, with the right experiment at the physiological level, you can connect it down to the biophysical level. But that becomes much harder when we want to explore how muscle behaves in a locomotor context. And so that's where we've had like the work loops come up before. So Josephson developed these based off of some of the sinusoidal analysis in the 1980s and 90s. And in this kind of experiment, you play in the strain pattern into an isolated or intact muscle. Sometimes you can do this in vivo, and you activate the muscle and you record the pattern of force or stress that results, and you plot those against each other. And so the area inside that curve is either work or mass specific work. And so you can characterize muscle function then by its mechanical work output. In this case, this would be a classic motor-like muscle because you get more force during shortening than during lengthening. But then following that, there was sort of this big appreciation that those first work loops, they actually tried to measure the conditions that would produce the maximum amount of work or power output. They weren't necessarily considered about what was actually created in vivo. And so when people started to look at the length and strain conditions and the activation conditions in vivo, we started to realize that there was a large variety of different kinds of work loops that a muscle could manifest. So you could have the brake-like muscle in red here. If the loop is reversed, it's the motor-like muscle in red. If the loop is reversed, it's a brake-like muscle in blue, or you can have these bifunctional muscles in gray. And you can even have this all be the same muscle just by changing when the activation occurs. You can get a change in work or power, and the same muscle can change. So this is sort of where we were at in the early aughts. But what's happened in the last 10 years is that there's sort of been a big appreciation that even these sort of steady state functions of muscle may not be where muscle really is under selective pressure to behave, because animals are perturbed all the time. And in fact, the perturbations oftentimes place greater demands on the different components of an organism than the steady state behavior. And so there's been a big push to try to do experiments that connect to the unsteady environment when there's perturbations. And that's quite tricky because muscle is buried way down in here. And so to go from the unsteady environment, you have to appreciate what the interaction is with the limb and the body and the other kinds of structural elements. And you have to take into account what's happening to the nervous system. So recently I got to write this review with Emily Abbott and Greg Sawicki, who are on the call as well. This is going to come out hopefully pretty soon. And we sort of identified 2 different approaches that people have taken. I'm just going to reprise those very quickly for you so you can see the structure that we're talking about. So we have to connect from the unsteady environment to the muscle. So we can start with a top-down approach. In the top-down approach, you can do things like I did in my earlier work, where you can record the EMG modulation, the change in neural feedback that an animal experiences as it runs over rough terrain. I did this in cockroach muscles. That's a little difficult to identify how an individual muscle is contributing to locomotion, because all of the muscles are changing simultaneously. So then you can take those patterns of neural feedback and you can play them back into an unperturbed animal so that you manipulate just a single muscle at a time. And in invertebrates, you can do this very precisely because each muscle is typically a single motor unit. So you can do this spike by spike with neural activation. And then you can record the length change that's happening to the muscle and the activation. And even before you calculate work, you can see that the same muscle under the same neural commands can do very, very different functions. And so we played in the exact same pattern of modulated neural feedback to this muscle in 3 different contexts. And in one case, it caused the animal to turn to the right. In another case, it caused the animal to not turn at all and just accelerate vertically. And in a third case, it causes the animal to turn to the left. So the context in which the neural feedback plays out, as good muscle physiologists and biophysicists, we sort of might appreciate this, but this is sort of a bit of news to the neuroscience community that muscle really matters. We can then try and understand the function, though, and this is the rest of the top-down approach. So we can actually take the strain patterns that we record out and play them back in through a work loop experiment. So this can be in an isolated muscle or an isolated joint, and you prescribe the strain changes, prescribe the activation, and measure the force. And in the cockroach case, just in this running case, what we can see is there's sort of a superlinear recruitment of work as you add neural spikes. And that we can actually unravel then that actually comes from a positive mechanical feedback coupling between strain and activation. So as the muscle is activated more, it produces more force, but that actually keeps the limb on the ground and in its shortening phase for a longer period of time. And that gives the muscle more time to produce force, which keeps the limb on the ground for even longer and gives it more time to produce force. So you get this superlinear rise in positive work because that is manifest only at the level of the whole locomotion because of how the muscles are interacting with each other. And so in this sort of top-down approach, we sort of started with a moving animal and zoomed into the work that the muscle could do. And the sort of future directions there is to sort of add systematic perturbations that help us further explore the patterns of activity, or connect it to multi-scale modeling efforts to try and understand, or multi-scale experimental efforts that I'll talk about in a minute, to try and understand the molecular mechanisms that are enabling this diversity of function under unsteady conditions. And I just want to point out that you can also do this all in vivo if you're lucky enough to work in a vertebrate system where you can add tendon buckles and sonomicrometry crystals like Manduka has with these great guinea fowl experiments. You can actually do a lot of this all in a freely running animal. And so there's a whole sort of suite of experiments that they've done now using this paradigm where a guinea fowl will drop down an unexpected or an expected perturbation down or up. And even before you record work output, you can start to see some of these same patterns in vivo. If you look at 2 different muscles, the lateral gastroc and the digital flexor 4, you can see in one case, the EMG patterns are changing and driving the change in force output, but the fascicle length isn't changing much. But in the digital flexor case, the fascicle length is changing a lot, the EMG isn't changing much, and you again get a change in work. So in one case, you have neural feedback mediating the change, and in one case, you have mechanical feedback mediating the change. And then when you look at the work output from all of this, you can first look at it at the joint level, and this led Manduka and Andy Bieliner and others to predict this sort of gradient of proximal to distal control, where the more distal muscles and joints do more of the modulation of work output during perturbation. So we're starting to get principles of how unsteady muscle function could arise depending on the location in the animal. And this can extend also to individual muscles now, looking at just the lateral gastroc. This is in the recovery step, in the actual perturbation step, before the muscle even has time to experience a change in the EMG response. It can already modulate its work all the way from being a negative brake-like muscle to a positive motor-like muscle, just because of the strain differences in strains that it experiences. So I hope I didn't slaughter Monica's past work here, but you can take a look at the papers for, I mean, there's a whole suite of these that are very nice to read if you want to take a nice afternoon sometime. The second approach that we can sort of take is a bottom-up approach. And in this approach, you start with the work loop and you can record the, so in a normal work loop, you would prescribe the length change that the muscle is undergoing. But instead of doing that, what you can now do is you can couple the muscle to a computer simulation that records the work the muscle is redoing and plays it through some mechanical model. And so this is like creating virtual reality for a muscle. It allows the muscle to interact with a virtual load or a virtual environment. And for one example that sort of Greg Sawicki and Ben Robertson have done is to use this to sort of pull out the contributions of tendon and muscle to the overall muscle tendon motor unit. To the muscle tendon unit work loop. And so you can record the force output here. And what you'll see is that the muscle work loop here in the shaded region and the tendon work loop here combine to produce a very different response in the overall integrated motor tendon unit. So using these simulated environments, you can really start to uncover how different components of the structural system are contributing to the overall work output. But you can also connect these to robotic systems. So in Greg's work, this might be a robotic exoskeleton where you're tuning joint impedance to the amount of work that the muscle is doing. But a really classic example now of this is Chris Richard and Chris Clemente's work, where they actually coupled an isolated muscle work loop experiment to a robotic flapping frog foot. And they could 3D print different frog feet of different morphologies. And by connecting, so that you would use the robotic system to prescribe the length and the force to prescribe the actuation on the robotic system. And this allowed them to look at what would happen if they scaled the foot morphology across sizes. And they found a nice little relationship that the way that hydrodynamic forces scale is very different from the way that muscle forces scale with size. And so there has to be this adaptive control of how a muscle behaves in order to keep behavior the same as the frog grows and changes morphology. And so we can use these robotic systems to couple the work output of a muscle to the environment that it's experiencing. And so just as the sort of future directions, people can then add sort of sensory afferents and try and close virtual feedback loops around the sensory control of the system. So adding a virtual neural environment. And we could take these all the way up to think about actually doing augmented locomotion in animals and humans. And this is some of the connection to human health that's going on now. So actually creating tunable exoskeletons that could interact with the biological system. So in this case, we've sort of, the approaches that we talked about here in the third approach, we're really trying to take the unsteady behavior and understand what the physiological changes in the work output and force production of the muscle was. But this doesn't yet allow us to connect to the molecular mechanism, which is what we sort of started out with when Natalie was talking. And so one thing that's also happening is we're starting to be able to do some really cool simultaneous experiments where we can try and measure what's happening at the molecular level at the same time as controlling the macroscopic level. And I want to talk just a little bit about this. I'm going to bring this, this video back up. Some of you may have used this in teaching. I know I do. I like this video because they get so much right and they get so much wrong. Right? So you actually see the myosin head like wiggles with sort of a faux thermal energy and everything. And so you can see this really nice interaction and they get the ATP and ADP cycle correct, but there's so much missing, right? This, as Natalie said, this is a really crowded environment. And just to give you an idea of size scale, there's about 20 nanometers here. ATP is 1 nanometer. So you're in an incredibly crowded environment just to get your energy molecules and to power cross-bridge cycling. And in fact, if you measure what's called the Peclet number, whether you can treat this as a continuum mechanical system or you have to treat each molecule as actually having finite size, then you're at a Peclet number of about 1 here. So you have these really interesting interactions that are happening here. But the other part of this simulation is that it shows the classic cross-bridge sliding, right? And just to show you the things that we haven't even considered, what happens in the other direction? What happens radially here in the up and down direction? It's just something that we haven't thought a whole lot about, but we know that actually myosin heads produce significant radial forces. So you could imagine just asking a sort of simple question to sort of frame how complicated this multi-scale question can be is, as you shorten a muscle, we know that muscle as a whole is isovolumetric. We knew that since 1666 with Jan Swammerdam, but what about the lattice? What about the myofilament lattice? Is the muscle shortens, do the actin and myosin filaments spread out? Or do they stay at the same length? And if they stay at the same length, that means the lattice is not isovolumetric and there's actually fluid pumping going on. So just this question of what's even happening in the orthogonal direction to sliding has been hard to tackle. But this is the kind of thing that I think we can now start to get at with these X-ray diffraction approaches. So this is, Tom Irving runs the BioCat line at the Advanced Photon Source at Oregon National Lab. And so what you can do is you can take your physiological prep, and because the actin and myosin are so well ordered, you don't need to crystallize the muscle in order to do diffraction through it. The muscle is already a living crystal. It's got such a regular pattern of actin and myosin that if you take your muscle preparation that's physiologically viable and you just drop it in this kilometer-long accelerator and you shoot the X-rays through it, you get a sort of very different view of muscle than many people have seen before. So this is actually the diffraction image of muscle, and this equatorial axis going across here corresponds to the spacings of the various actin and myosin containing filaments. And the meridionals here, the orthogonal axis, corresponds to the helical repeats of actin and myosin and the location of the cross bridges along here. And so this has actually been around for about almost 30 years now. But what's really cool is that just statically, this wouldn't tell us a lot to connect to muscle function, but we can now do it dynamically. So we can, while we're doing a work loop preparation, we can shoot the X-ray through, get the diffraction images. And we can do that now at a scale where we can do it every millisecond. So we can collect 1,000 to 3,000 diffraction images every second and get a sort of time resolved high-speed movie of at least what the aggregate properties of the structural interactions of actin and myosin are, and sometimes maybe the other filaments in the system as well. And so just as an example of what that can do, so here I'll just show a movie. So this is a contracting Manduka flight muscle, slowed down about 40-fold. And you can, if you take a close look, you can see these peaks coming in and out of alignment, so changing their intensity and their spread. A lot of the business is actually happening in this central region. You have to make it a lot less bright in order to see that. So here's a zoomed in on that region. And you can see these, this first fringe here corresponds to the orthogonal distance between actin and myosin in the crystallographic plane. And you can see that it'll come in and out of this sort of alignment where it spreads out or gets more intense. There's also a small amount of change laterally that corresponds to the movement of those filaments. And so these kinds of experiments, the muscle, you can actually have it, you can be having it measuring force. You can even have it inside the animal. You can put the flapping insect into the X-ray beam, but sometimes it's better where you can control the length and measure the force. And what it allows us to do is it allows us to connect the nanometer scale changes of those filament spacings to the macroscopic scale at the same time. And so just an example of what this allows us to do, there was sort of these two muscles in the cockroach leg that have sort of created a mystery for a long time because they have pretty different work loops. One is a brake, one is a bifunctional motor. But every other way that these muscles have been measured, they've been identical. They have the same force length curve, force velocity curve, the same activation, the same moment arm, they share the same motor neuron. So they should be doing the same thing, but they're different. And the first sort of difference that we've ever been able to find here is that one of them has a nanometer tighter lattice spacing. So one of them, the myosin head, is further away from the actin than this one, which is more constrained. They're very creatively named muscle 178 and 179. But as the muscles contract, this one pushes out and this one stays at its same thing. So this one undergoes a radial transient. In this case, the lattice spacing moves in one muscle and doesn't move in the other muscle. And so we get these interacting scales that may lead to the difference in work output here. So those are sort of the 4 points that we wanted to, that Natalie and I wanted to make. I'll let Natalie summarize really quickly from her words, and then I'll do on mine, and then we can get to the brainstorming session. So Natalie, you want to summarize on this slide?
Sure. Just hit all the buttons. There we go. I can't deal with someone else animating me. It's weird. Yeah, so I think from mine and Simon's discussion about this, some of the big conclusions that I came to was this thing that I've been thinking about for a long time in terms of at a muscle level, how do we get from this kind of standard simple cross-bridge model up to something that's relevant to movement? And I think the things that we struggle with, and we've tried to talk about how we're developing, is being able to conclusively measure or infer what's happening down at this small scale. And I think Simon showed some really nice kind of new biophysical approaches that are happening. But then maybe one of the next big steps is how do we do this in a way that is relevant both to all of the interactions between muscles? So how do we take into account all of the cross bridges interacting? How do we think about them interacting with titin and with fluid and with connective tissues? So how do we make those very small scale measurements in a way that's relevant to what an animal does? And then I think certainly in my head, we're at this point where we understand that there's all of this structural complexity. And I feel like everywhere I look, it does something important in terms of in vivo muscle function. So I think kind of refining this to understand what is actually important in terms of these different structural elements in order to make better models or have predictive understanding of muscle. And I think at the moment we're kind of just doing everything and looking at everything. And so how do we refine that to what's most important?
And I think we wanted to say that in this approach, you know, with this talk, we were really excited to do this, but we could only sin by omission, right? We could only leave out other things. So if you feel like we left out the most important experimental approach that you feel really passionate about, great, please bring it up in the brainstorming
Please
session.
tell us.
And we apologize. We know that there's a lot of interest in shape and size, and we haven't touched too much on that, on aging and changes in the structure of muscle. But just to launch us, we wanted to say that we sort of have this Humpty Dumpty problem, right? We have a lot of techniques to take things apart in muscle, but it's very hard to synthesize the ideas back together again. And there's this trend to sort of bridge experiments that show molecular mechanism and function, but they're very hard to do, and there's very limited context in which they've been done so far. So we really need to try and measure, observe, control, and manipulate in a context now that links to physiological function. I think that's a lot of the hope and promise of the comparative approach. And I think one of the things that's really been lacking actually is these larger scale efforts where we sort of tend to be a pretty disparate group community, in part because there's usually only one of us at each university. There's very few of us on this call that have another colleague at the same university. And I think that's what's so great about the CNB and several other approaches now, that we really need sort of community efforts that are aligning and trying to tackle similar problems from different angles, rather than all spinning off into our own directions. And that's a criticism as much as for me as anyone. But ultimately, we don't think muscle has lost its allure. It's still the tissue with the most amenable to this sort of principled multi-scale understanding. And that's what came from the original work from sliding filament idea, connecting the biophysics of the molecules all the way up to the macroscopic function. And it's still one of the hardest tissues to emulate, but remains an aspirational target for bioinspired design. We still really want to be able to understand and emulate muscle. And our sort of last point is that its proper function is still also one of the central challenges to human health. And this is something where we think a comparative approach can really be beneficial. So with that, those are our acknowledgements of the people that we've worked with on various parts of leading to this. And we'll turn it over to Greg and Monica.
Thanks, Simon and Natalie. That was a great overview. Let's see, I'm just going to go ahead and share my screen now again. While we get ready for the breakout sessions, do we have any quick questions for the speakers before we go? Anyone? All right.
And one thing, Monica, I was going to mention, if after the breakout sessions, when we all come back together, is also a time when we can ask questions of the speakers. Maybe the breakouts will get some questions that you didn't already have on the forefront of your brain, and then you can ask when we come back to the discussion.
Yeah, and if we go a little, we might be running a little bit over time. So what we'll do is we'll probably just eat into the break rather than cutting the discussion short. So we have a half an hour break scheduled. So we'll probably just roll into that a few minutes over time so that we can make sure that everybody has a chance to discuss in the breakout groups. And then we have plenty of time in the group discussion as well. So I'm going to go ahead and share my screen again. And so we've had a really nice overview of the different experimental approaches that are currently being used and have historically been used to understand muscle function. So I'd like us to now break up into our groups and talk about what are the biggest challenges to integrating across organizational scales. what are the key gaps in knowledge, and what are the tools and approaches that are most needed to fill them. So our task in our breakout groups is to create a list of 3 experimental aims or approaches that are most needed and discuss those as a group. And so hopefully we'll come up with lots of different ideas that we can then pull together and discuss. The idea here is that, you know, by bringing in lots of different expertise, hopefully we'll come up with some new collaborative ideas that will help us bridge these interdisciplinary boundaries and encourage more collaboration across institutions and research groups. So I'm going to go ahead and try to break us out into breakout groups. I'm going to stop the live stream for now while we're in our breakout groups. And then the live stream will return when we start our second set of keynote talks, which will be at 12:30 p.m. Eastern Daylight Time. So I'm going to stop the live stream now.
We're about to get part 2 rolling here. I thought we had a really nice morning. It's really cool to see how the discussions all coalesce onto a few, maybe 5 really key points that I think we'll be able to summarize and share with the broader community later today or later this week. I'm going to share screen now. And we'll get started with the second part of the day. Let's see here. Hopefully everybody can see that. Module 2. So we're in the morning, we focused a lot on multi-scale aspects of muscle and how it functions all the way from molecules up to whole body. And in the second half of the day, we're really going to turn our focus a little bit more onto modeling. How best do we describe what muscle does in computer simulations and with mathematical theory? And to get started there, we have our second keynote of the day. It's a tag team effort by Friedel de Groot from KU Leuven and Madhu Venkadesan from Yale. And once they're done with their talk, which should take about a half an hour, we'll switch back into our breakout session mode again, where we'll focus on outlining best practices for validating models against experimental data. Okay, so I think without further ado, I'll turn it over to either Friedel or Madhu. I'm not sure who's starting off, but I'm going to stop sharing my screen now and turn it over. And you guys should have the stage whenever you're ready.
I will start off. I'll do a share screen and such. All right, can I get a, yep. All right, so our objective, by our, I mean Friedel and my objective, is to show examples really of different types of uses of models or different types of models and how we think about muscle models in many different contexts and make use of them too. I'm going to go first. The very next slide I'm going to show was put together in the last 2 minutes, motivated by some of the discussions and some of the morning talks. So forgive me for misspellings in that. I haven't even seen that slide yet. But so my topic is going to be just a few lessons that I think I learned from the soft matter physics community and how to use that to approach muscle. A lot of the work, as you can guess, of our own results that I'm going to show are done by students in the lab. I want to acknowledge Koi's important contributions and Nilima. Their contributions was not just the work, but the number of conversations that led to intellectually new things for me. Some of the work I will talk about are in either published papers or not yet peer-reviewed preprints. And towards the end, I'm going to make some references to a real motor behavior, and some of the data for that reference came from this paper. Okay, let me start by why do I care about soft matter physics? Very loosely, it's soft, it's some matter, but it's a little bit more than that. And for that, I'm going to turn to inspiration from Pierre Digen's Nobel lecture, Pierre Dijen, as many of you know, got a Nobel in physics for his work in theoretical physics and viscoelasticity in particular, and renormalization, group theory, and so on. But he has a very telling statement in his lecture where he tries to very early on define soft matter. He says a very mild chemical action induces a drastic change in mechanical properties, a typical feature of soft matter. I mean, to me, I did not grow up in the soft matter world. I came from thinking about biomechanics and muscle before I read this lecture, and I said, hey, he's describing muscle to me. He never really talks about muscle, and not surprisingly, later in his life, he did work on trying to build a muscle-like actuator driven by his own thinking of soft matter physics. I want to acknowledge a few funding sources. That's a private donor who funded some of the work. PEB is a graduate program in Yale for people, graduate students and faculty who are confused about which world they belong in. And I wish to acknowledge support from NIH and NSF as well. So let me dive, and I guess I should say something about that. If you point your phone at it, it'll take you to my lab webpage, so you don't have to type. If I want to summarize the things that I will bring up in my talk today, can be summarized as a set of keywords. I'm going to talk to you about rheology of muscle and introduce very closely related ideas of either dynamic stiffness or complex modulus, and I will define these more precisely as I go along. And the contexts in which and behaviors in which I want to think about those are how materials respond to perturbations, especially things that are active like muscle. And the notion of stress relaxation time becomes important in that. And really, the way I would like to think about muscle is it's matter, but it's a tunable matter. But there's an external signal that can tune the type of matter it is. Okay, now is the promised bad slide or hurriedly put together slide. And the purpose of what I'm going to, before I dive into very specifics, is my very sorry attempt at trying to classify the world of muscle models. Of course, I've left out a lot. I've presented a very subjective, opinionated cross-section. And here is the cross-section. And I want to call them models and theories of muscle. I think very well-drawn pictures or cartoons are a model. And they are quite useful, informative, help us understand a lot of things. And biology has done exceptionally well with such means of describing processes. That's not the class of models I'm going to focus on. I'm instead going to focus more on quantitatively precise models. Note, I call it precise, not necessarily accurate. So you can have very imprecise pictures that are quite accurate and precise models that are quite inaccurate. So you have all possibilities, and I'm interested in the quantitative nature of those. Of course, there are macroscopic ones, and we are familiar with a bunch of macroscopic ones. Things like Hill model of muscle or short-range stiffness is a property of muscle, but there are modifications of Hill models to incorporate short-range stiffness, and I would call those macroscopic models. Tom Roberts and many others, Natalie was talking about it too. Passive tissues play quite a big role in how muscle behaves, and those are really macroscopic phenomena we're paying attention to. There's microscopic phenomena. A lot of the drawings are of the microscopic phenomena. And the microscopic phenomena I will classify as first deterministic models, where we are not paying attention to temperature-induced fluctuations. So concepts like entropy are not really part of that thinking. And examples, are modifications to very classical muscle models that have come about are currently in that trend. The winding filament model of Titan from Kisa is primarily a deterministic model. There is a notion of stochasticity in these models, but there's not exact accounting of entropy in these. What Simon was talking about is another example of a very important modification, which is paying attention to the lattice structure itself. Tom Daniels has, for example, done work on thinking about the pumping action because of the relative motion between the fluid and the lattice, because the fluid stays roughly incompressible, but the lattice is not, and that has consequences. So these are examples of deterministic thinking about muscle, which of course means I will talk about stochastic thinking, and that's where I will spend a little bit more of this slide on. Okay, how do I think about stochastic models? This is my opinionated classification of stochastic models. There are thermodynamics associated phenomena and the level of spatial detail in those phenomena. So the spatial detail can go from lumped models. An example is Huxley's model of cross bridges with or without tightening, as it correctly needs modification today, or spatially explicit ones, where you actually pay identity to the fact that not all the myosin motors are sitting on top of each other. And that affects phenomena such as cooperativity, which Sam Walcott has published some nice papers on. Then stochastic, so there is thermodynamics, but I can assume that you're in thermodynamic equilibrium, so that fluctuation dissipation is exactly satisfied in these systems, or it is out of equilibrium and beginning to relax towards equilibrium. Pretty much all statistical mechanical theories we have today live in this equilibrium. Non-equilibrium theories are very rare in general, and in muscle even rarer. I don't even know how to come up with a name for examples of non-equilibrium theories besides an example of a citation. So Caruel et al. Have published multiple papers thinking about populations of myosin motors, but they're out of equilibrium behavior and not, so things like rate functions and so on make sense only in equilibrium thermodynamic systems. In green, these are not models, these are experiments. But to me, the sort of insights those experiments give are really a way of thinking about muscle. So spontaneous oscillatory contractions is the name SPOC that Ishiwata et al. Have. Dan Rassier has done some really nice work looking at how spatially varying the behavior is. And these are experimental systems. They are fundamentally out of equilibrium. So equilibrium theories do not help us understand what's happening in those systems. And I think that is the real world of muscle models where fundamental insights are missing. Our theoretical tools are also missing. There's a whole venue of opportunities. That's not to mean that any one of these is solved or useless. There are lots of pieces to be resolved there and incredible utility of those models. Macroscopic models are very popular with people modeling whole body behaviors and doing optimal control because of their computational simplicity. Deterministic microscopic models or equilibrium stochastic models, I would argue, are also quite tangible computationally today. Combinations of GPUs or approaches like Zehelak's modal decomposition, you can convert PDEs into ODEs, and we can deal with ODEs and even PDEs quite well in near real time these days. But the minute you enter non-equilibrium, forget about computation, even the models are on tenuous ground right now. Okay, that's my attempt at classification, and I'm going to hope that this is somewhat provocative so we can have heated discussions. Let me then dive into what I would like to tell you about, which lives somewhere between the macroscopic and the stochastic world. The dotted line I've not drawn. That's roughly where my attempts are going to live. So let me start with an overview of what my talk will be. I want to tell you about a rheological view of muscle. So therefore, I'll tell you what do I mean by rheology in this context, and use that to understand what are cross-bridge models even capable of. Based on that understanding, I'm going to try to develop a prediction, a hypothesis for muscle as a tunable medium. And in this picture, really, we know that dead muscle has some sort of elasticity to it. Resting muscle, what I'm calling passive tension, as the earlier breakout session brought out, there are at least 3 different names for this in different communities. What I mean is the passive tissue, depending on its stretch, will have some tension, but if I try to perturb it, it will respond with an additional reaction. You can think about similar components for the active energy consuming part of the muscle, which need not be all in cross bridges, the active response. And approximately, the whole thing is in series with the tendon, depending on what the tendon is, but this is an approximation. We often have, and in the back of our mind, data on a whole muscle that we would like to use. The very highly controlled experiments tend to be at the scale of single fibers, and beautiful pictures are often generated by experiments at this scale, and maybe the microscopic understanding is generated at that scale. This is an example of this type of cartoon I was talking about. So this is to refresh your memory on what muscle is. Muscle is myosin at the cross-bridge level, myosin undergoing these cyclic changes, but muscle is not all that. A lot of these are assembled in a very spatially ordered manner, including missing components in classical pictures like Titan, which then there's a whole cascade of arrangement of tissues that leads to whole microscopic behavior. I am going to not pay attention to any of these details when I take the rheological view, because I'm going to take the Hill approach of there is a lumped black box. What can we understand about this black box by recognizing that it broadly follows principles of soft matter? So I'm going to simplify the picture down. So I have a muscle, and the type of experiments I would like to think in the back of my head is that total length of that muscle is going to be varied in some time varying dynamic way. I'm also going to activate it, but the activation need not be constant. That could also be time varying. And naturally, in this system, I will record forces that are time varying. One manner of thinking about it is this is an approximation that there are classical Hill type forces, but on top of it, there is a perturbation response. So like Simon was talking about, the force length, force velocity pictures are really quasi-static. They don't tell me too much about how muscle as a tissue responds to perturbations. And the perturbation response is itself modulated by activation. So hopefully a provocative statement, which I'm going to make. I think we as a community are hung up in a not productive way on forces and what is the force of muscle. I think the real question is, what is the muscle as a material? What is it doing? In other words, what is the transformation between length and forces, and how does that depend on activation? How does that depend on what has happened to muscle in the past? Force itself is then a side effect, but an important side effect of the material property of muscle. How do people study this in passive materials? Let me use this is not even a paper. I think this is published as some sort of a popular article, but I would point you to it as a very nicely written article on soft matter rheology and its uses. Let me walk you through what he's doing here. So what Dave Weitz is walking us through is, if you take an elastic solid and subject it to sinusoidal strains of some fixed frequency, and it's a sinusoid, and that's sort of important here. For a perfect elastic solid, the response will also be sinusoidal for small oscillations and be exactly in phase with the strain. If I had a classical Newtonian fluid, linear viscosity, strain and stress will be exactly out of phase. That's because in a viscous fluid, the stress responds to velocity of the strain. The strain is like the cosine, it's like the sine of time. Then the velocity is like the cosine of time, and that's why they're exactly out of phase. Of course, most materials are neither. They are viscoelastic, for want of a name. And that means that stress and strain have some phase relationship that's not exactly in phase or exactly out of phase. So they have some phase relationship, and the degree of phase offset tells me something about whether it's a solid-like or a fluid-like material. We can translate these ideas to muscle in the following approximate way. The math can be treated as reasonably accurate and exact, but the words are my own and my own interpretation of what those terms represent. Right? So I can think of the length as having some baseline length and perturbations around it. Right now, I'm not thinking of the perturbations as sinusoidal. There are some perturbations. In response, I will see force oscillations or fluctuations. And I'm going to call that as some baseline term plus an active perturbation response, which includes things like the force length velocity. It's not excluding those, or it's not on top of those. It's all part of one big bucket. Can I request a time check? I just realized that I don't have any sense of when I started.
I think we should be switching over to the other speaker within the next couple minutes.
Okay, I'm in trouble.
Okay.
So let me proceed and only present briefly touch upon what research we have there in the preprints I pointed you to. So the idea is, if my oscillations are sinusoidal, my force response using a Fourier decomposition can be thought of as a sum of sinusoids. And the linear picture is that, to leading order, the force response is also sinusoidal, and the sinusoid has an in-phase and an out-of-phase component. And the in-phase is representative of the elastic behavior, and the out-of-phase is representative of the viscous component. And so they're called the storage modulus and the loss modulus. So, but these moduli depend on the frequency at which I oscillate. And of course, any general length perturbation itself can be thought of as a sum of sinusoids. So understanding perturbations that are sinusoidal and their responses is quite important. If you take simple systems like a spring and damper in series, even that system can show complicated looking behaviors like stress relaxation. Stresses build up and relax over time. And that time scale is just a function of two parameters, which is the stiffness and damping, as simple as it gets. If I look at the dynamic modulus, which is the sum of squares of the two moduli square root, the dynamic modulus depends on frequency in some manner. Even though I have constant stiffness and damping, this dynamic stiffness, or dynamic modulus, has a frequency dependence, and the storage and loss moduli vary with frequency. So it's this picture of a dynamic modulus that I would like to use for understanding muscle. The first result that we have, and I will point you to our, this is not even a preprint, it will become a preprint in some time, is that if you consider various forms of cross-bridge models, 2-state to 5-state, all of them essentially behave like a spring and a damper, with notable differences in the region where they transition between a purely elastic solid and a purely viscous. There is some experimental support that that's the sort of behavior you see in real isolated fiber experiments as well. But again, there are notable departures from that theory, which require a little bit more thought, and its consequences are to active motor control to be decided. You can also take this rheological view to an extreme and have a geometric viewpoint. And the geometric viewpoint, again, starts with passive materials, where if I oscillate in length sinusoidally and plot the forces, for small oscillations, I see these ellipse-like figures. For large oscillations, I see more banana-like figures. But the ellipse can be thought of as coming from the in-phase and out-of-phase components. So the storage and loss modulus govern the shape of the ellipse. But in muscle, we have this additional thing called activation, which changes what type of material it is. So you can, one hypothesis or we have is one way to think of muscle is that the force response to sinusoidal oscillations comes from a family of responses where the activation is simply interpolating between those responses. So you can splice these responses together to try and construct a complete response. So here is an example. If, say, a muscle with low activation has this ellipse, but a muscle with high activation has that ellipse, and I try to construct a work loop of switching between ellipses, then that's the shape of the work loop I will construct. And that construction is simply a consequence of the assumption that activation is switching between these. This is not a complete theory, and there will be exceptions to this. And the type of exceptions are hopefully places where we can dig to find out why is activation not a simple extrapolation. I'm going to jump through because I would like to show only two small things. One is that what's the value of some such theory? One potential value is we can construct all possible shapes to be described by a parameter space. And this parameter space has no dimensions. It's a dimensionless space. And therefore, pretty much under that theory, every possible muscle, when I subject it to a work loop, it's a work loop experiment, is going to adopt shapes from this space. If you now ask, what can a hill model do? A hill model, because force length curves have a very distinct plateau in the middle, mostly occupies only this region of the space. But as we know from many work loop experiments, muscle can span a lot of other areas. So I'm sort of turning work loops around on their head. They were maybe originally meant to mimic oscillatory motions as sinusoids, but I'm saying doing a sweep of sinusoids can help you understand a more general property of muscle. And these are examples of real work loop data where by eye, we try to fit ellipses to it. Sometimes the ellipses make very obvious sense. Sometimes the ellipses have negative slopes, and there are questions of can you get negative slopes and negative storage modulus in that muscle. That's at least a provocative hypothesis. All right, so I'm going to finish by only pointing out that this is not to say that there are no new phenomena, and I will point out one phenomenon in the context of a pretty straightforward behavior. I can throw at that speed, I'm sure. I'm kidding for those of you who don't know me. I can barely throw a third at that speed. But when people throw very fast, your muscles can experience enormous strain in a very short period of time, and effectively dissipate stresses when you have a fairly low activity in the muscle. On the other hand, as work of Eric Perot, Tom Sandercock, many people tell me that you can stand, you can do many things you do because muscle can maintain stresses for hours. So muscle can, in certain circumstances, dissipate stresses in milliseconds or support stresses for thousands of seconds. There's a million-fold change in an intrinsic timescale. And current cross-bridge models, the best that I can think of, can account for about a 1.2-fold change in this timescale. So we talk about connecting scales of length scale. I would like to point out that connecting time scales is yet another problem that's just pending to be solved. I stop here, and I have overshot by an indeterminate amount of time. Thanks.
Okay, so then I'll take over. I'll share my screen. Okay, so I want to start with also thanking Greg and Monica for organizing this amazing workshop. I'm really glad I can be part of it. And so my talk will be complementary to Madhu's talk, as I will be focusing less on muscle modeling itself and more on the use of muscle models in simulations of whole body movement. So I believe that simulations are a powerful tool to understand principles of movement, but the high computational cost of predictive simulations has long limited model complexity. Studies based on complex 3D muscle-driven models were typically based on a single or just a few simulations. But new computational approaches now enable rapid simulations based on complex muscle-driven model. And so here I will introduce whole body simulations and illustrate what they can and cannot yet teach us about how muscles shape whole body movement. So let me start with introducing the framework. So the model of the musculoskeletal system describes how muscles transform excitations into muscle forces, how these forces are applied to the skeleton, and how the skeleton moves under the influence of muscle and external forces. And for whole body simulations, we also need to model motor control. Very often, and that's what we do as well, it is assumed that motor control is optimal in some sense. For example, by minimizing metabolic energy. And we can then find the optimal neural controls by solving an optimal control problem. And this optimal control problem has the following form: minimize movement-related costs subject to muscle and skeleton dynamics. But solving this problem is hard due to the nonlinear and stiff equations describing muscle and skeleton dynamics. Stiffness reflects that a small change in the controls might result in a large change in the states. So recently, new computational approaches have been used to solve this optimal control problem. And we improved the computational efficiency by combining direct collocation and implicit formulation of muscle and skeleton dynamics with algorithmic differentiation. And we are now able to simulate human gait based on complex 3D muscle-driven models in about half an hour, instead of about 13 hours, which is a reduction in computational time by more than a factor of 20. And I think this opens possibilities to explore the relation between muscle function and whole body movement. So we'll come back to predictive simulations, but I want to start with inverse dynamic simulations. We use the exact same computational tools to analyze measured movements. In these cases, we know the movement and we want to compute the underlying muscle activations and energetics. This requires us to solve the muscle redundancy problem. Typically, we do so by minimizing activation squared subject to muscle and subject to muscle dynamics, and then a constraint expressing that the muscle forces should produce the experimental joint torques. In such inverse approaches, muscle dynamics is typically described by the hill-type muscle model that we have been talking about before. So in this model, the tendon is represented by a nonlinear spring in series with the muscle. And the muscle is represented by a passive element in parallel with a contractile element. The force produced by this contractile element is a function of muscle activation, fiber length, and velocity. And the dependence of force on length and velocity is described by analytical functions derived from the quasi-static experiments that Natalie and Madhu mentioned. Those hill-type models are attractive because they are relatively simple. And in addition, they can be scaled to any specific muscle by only 5 parameters. And for humans, these parameters are readily available for a lot of major muscles. So we use this inverse approach to study energetics of forefoot and rearfoot running. And we found that metabolic energy consumption was similar for both foot strike patterns, both at the whole body and triceps surae level. But here I want to draw your attention to the changes we had to make to the generic muscle parameters. So we used a generic OpenSim model, and we scaled it to the subject's dimensions. But first we had to multiply the maximal isometric force of all muscles by a factor 3 to obtain submaximal muscle activations for running at 14 kilometers an hour. This is not very realistic and might indicate that the hill-type model does not accurately describe muscle mechanics during dynamic movements. Anyways, we performed simulations with this extra strong model and validated our simulations against gastrocnemius medialis fiber lengths measured with ultrasound. We found that simulated fiber lengths were very different from measured fiber lengths, but when we reduced Achilles tendon stiffness by about a factor 2 with respect to the generic model, simulated and observed fiber lengths were in good agreement. So altering Achilles tendon stiffness also had a large influence on the simulated triceps surae energy consumption, which is not surprising given its dependence on fiber length and velocity. So in the running study, we reduced Achilles tendon stiffness equally for all subjects. We also developed methods to estimate Achilles tendon stiffness and triceps surae parameters by tracking motion capture, EMG, and ultrasound data. Individualizing muscle tendon parameters improves the fit with measured data with respect to using generic parameters, both for calibration trials at low walking speed and validation trials at high walking speed. But again, it might be more useful to look at the limitations of the approach. Typically, we did a bad job at fitting experimental fiber lengths, which are in blue here, around stand swing transition indicated by the vertical line. And the fit was even worse for pennation angles. And this might be due to the underlying modeling assumption that muscle width is constant in a hill-type model. We know it's not, but I feel we don't have a good alternative for this assumption that can be integrated in a hill-type model without relying on more complex 3D models. So I also used this inverse approach to study reactive standing balance in collaboration with Lee Natting. We've already heard that the hill-type model is based on quasi-static experiments and might not well capture the reaction to perturbations. In this study, we focused on the first 50 milliseconds of the response to platform perturbations, where alterations in muscle activity are absent due to sensory motor delays. Using a hill-type muscle model, we could not explain experimental joint torques in black here during the initial response, even not when we accounted for large levels of co-contractions. So when we added a phenomenological description of short-range stiffness to the model, we could explain the observed joint torques. And so this shows that we really need those history-dependent properties of muscle to describe how the muscle acts in like unstable environments. Or not sure what's, okay, so our forward simulations confirmed that short-range stiffness limits joint movement during the neuromechanical delay period. So we think that using muscle models that account for short-range stiffness, along with other aspects of history-dependent muscle dynamics, might be important to advance our ability to simulate inherently unstable human movements. So inverse approaches are a useful tool to study muscle mechanics and energetics underlying measured movements, but they do not allow us to study how muscle properties shape movement patterns, and therefore we have to rely or to resort to forward simulations. So let me start with forward simulations of human walking. In this case, we imposed gait speed and either symmetry or periodicity of the gait pattern. And we found a physiologically feasible gait pattern when minimizing a weighted sum of metabolic energy rate, muscle activity, joint accelerations, passive torques, all terms squared per distance traveled. And here you see this, the resulting gait pattern. So the same modeling framework also predicts key features of human gait with varying gait speed and muscle strength. So when we increased gait speed, the model predicted a continuum of walking and running gaits. And when we decreased hip actuator strength, the model adopted a compensated Trendelenburg gait as seen in patients. This gait pattern is characterized by hip circumduction and trunk sway. The framework is equally applicable to study animal locomotion. So our collaborators at the Royal Veterinary College, John Hutchinson and Peter Bishop, used the framework to study maximal height jumping in the Tinamou. Modeling muscles influenced jumping kinematics, but even in the absence of muscles, the simulation predicted a counter movement maneuver. The simulations also allowed to study the influence of muscle strength on jump height. Jump height was most sensitive to ankle actuator strength and less to knee or hip actuator strength. So I hope that these examples illustrate the potential of predictive simulations, but I also want to focus or to point out some of the challenges here. So notwithstanding the huge advances in computational efficiency over the last 2 decades, there remain computational barriers to using complex muscle models. Van Soest and colleagues showed that CPU times were 10,000 times higher for a Huxley than for a Hill-type model. So they performed whole body simulations with a quite simple model with 6 muscle, where maximal jumping simulations with both a Hill-type and a cross-bridge model. And in their case, they found that both models performed similarly, but I think that might be due to the use of very simple activation patterns. A second big challenge is our limited ability to personalize model parameters. So we aim at using predictive simulations for personalized treatment design, but creating those personalized models is hard. And typically we compensate for a lack of experimental input by making assumptions about how pathology affects muscle tendon properties. And it's yet not clear how those assumptions influence our predictions. Thirdly, the simulations I showed are not robust against uncertainty due to sensory motor noise or external perturbations. We, but also others, such as Anne Koelewijn and Tom van den Bogert, who are present here, have been extending the available tools to simulate movement, to simulate movements that are robust to perturbations. However, the computational cost of these tools still limits their use with complex models. And in addition, both experimental work and simulations have shown that simulating robust movements requires models that capture the history dependence of muscle dynamics. And then finally, I feel we need more detailed motor control models. And I want to end my presentation with an illustration of how motor control and muscle mechanics are intertwined. So, as I mentioned before, one of our major aims is to use predictive simulations to improve treatment planning in children with cerebral palsy. And the majority of those children has spasticity, and it's poorly understood how this influences their walking pattern. And this is not surprising, as we even fail to understand the mechanisms underlying the response to passive single joint movements as imposed during clinical tests of spasticity. So the pendulum test is such a clinical test. In this test, an examiner extends the leg of a seated patient and then drops the leg. The resulting lower leg movement is recorded. And a key kinematic feature in spastic individuals is a reduced first swing excursion. So I performed a modeling study in collaboration with Lina Ting to investigate the mechanisms underlying the reduced first swing excursion. We demonstrated that we could capture the reduced first swing excursion by modeling short-range stiffness and its interaction with muscle tone. So short-range stiffness is dependent on muscle tone, which is known to be increased in spasticity. And short-range stiffness is especially high after the muscle has been held isometric, and so therefore there might be increased resistance against movement during the first swing after dropping the leg. We then set out to collect experimental evidence for the modeled mechanism. Short-range stiffness can be reduced by moving the leg before releasing it, as prior movement has been shown to reduce short-range stiffness in muscle experiments. We found that moving the leg before dropping it increased the first swing excursion in both healthy children and children with cerebral palsy. And with this, I want to wrap up. So I think we now have new computational approaches, and they have made it easier to account for muscle dynamics in whole body simulations. So all our tools are open source, but in addition, also the OpenSim team at Stanford released MoCo, which has similar capabilities. So, and might be a little bit easier to use, although it also might be a little bit slower than our tools. So we know that the Hill model is inaccurate, yet it is commonly used, and that is probably because alternatives are not readily available or computationally more challenging. So I feel that the alternative muscle models are often not yet scalable based on a limited set of known architectural muscle tendon properties. This might change soon as we, but also others, including Matt Millard and Anne Koelewijn, are working on this. So, but notwithstanding the open challenges I discussed, I think simulations can be a powerful tool to study movement, especially when we complement them with experiments. And with this, I want to acknowledge my team, as well as my collaborators and my funding.
Okay. Thanks, Friedel. Looks like people are still with us. Thanks for that session. I am clapping. Everybody should too, even if you're muted. I think Monica and I were chatting a little bit just at the end here about how to handle Q&A, because we didn't, I guess, do a great job of incorporating time for group Q&A. But I think what we can try is we're gonna head back to the modules now, and then what we'll do before this, after the day and before the spatial chat social hour, we can all hang out in Zoom if you'd like, anyone who wants to, and we can open up the floor to all the speakers maybe and have a Q&A that goes as long as anyone wants to hang. Okay, so I'm gonna try to share my screen again just to give some prompts, give you guys reminders about what's happening in the next breakout session. And then it looks like Monica's gonna have to reassign some folks. So we'll go through the same rigmarole before, where if you're not assigned, Monica will do a roll call, and if you could just say which group you were in the first time, you're going to go back to the same one. let me quickly share screen here.